All right, welcome everybody. We will wait a few seconds as the virtual audience filters into the seminar room. So. All right, so welcome everyone to the Cary Institute of Ecosystem Studies' weekly scientific seminar series. We have a hybrid meeting today with some people in the audience here at Cary and others online. So welcome especially to our hybrid audience online. For those of you online, you can put your questions in the Q&A and we will get to them after the talk. And if you have any technical issues, please put them in the chat. So I'm delighted to introduce this week's seminar speaker, Camilo Zalamea. Dr. Zalamea is an assistant professor in the Department of Integrative Biology at the University of South Florida. He completed his bachelor's and master's degree in biology at Universidad de los Andes in Colombia. And he then completed his PhD in a joint program between the University of Montpellier in France and Universidad de los Andes. Before joining the department at South Florida, he did 2 postdocs at the Smithsonian Tropical Research Institute in Panama. Dr. Zalamea is a tropical plant community ecologist who focuses on plant microbial dynamics to understand the maintenance of biodiversity, especially in tropical forests. To answer his questions, he uses a combination of field, greenhouse, and laboratory studies, including molecular techniques. And I'm sure we're going to see a variety of his approaches today. Specifically, he studies how soil fungi shape tropical tree communities by either serving as a pathogen or facilitator of seed germination and tree survival at the early seed stage of plant demography. He also studies the degree to which tree species and microbes specialize on each other and on particular soil nutrient conditions. So I met Dr. Zalamea in Panama at the Smithsonian Tropical Research Institute many years ago, where he's a research associate, and he did one of my favorite studies on how different tree species specialize on soils of different phosphorus fertilities, which I really love. I always enjoy speaking with Camilo and find his work fascinating. So I look forward to your talk. Please join me in welcoming Dr. Zalamea.
Thank you, Sarah, so much for this very kind introduction. So I was told that people will hear me better if I don't use this mask, so I hope this is okay for people in the room. Well, again, thank you so much for this invitation. I'm so happy to be here talking about seed-microbial interactions and a little bit of what I do as a scientist. So just as a little reminder, I'm an assistant professor at the Department of Integrative Biology at the University of South Florida. I'm also a research associate at the Smithsonian Tropical Research Institute. And here you have my website, my email, and my Twitter handle if you want to get in contact after this talk. A little bit about my experience. So I find myself as a tropical plant community ecologist, and I am particularly interested on pioneer trees. And pioneer trees are very interesting for several reasons, but I will try to show you some of those later on in this talk. I am also interested on niche differentiation of tropical trees, and I am particularly interested on soil resources and microbial interactions in the soil and how those can let us understand how species distribute in the landscape and how species partition their niches in tropical lowland forest. And I am also interested in plant microbial interactions and in particular soil-borne microorganisms. And I guess they are important because they can tell us a lot about plant demography. And in particular today, I will be talking about seed microbial interactions and how those interactions can let us know about seed demography in particular. So let me start this talk by saying that obviously the work that I will be presenting today is not only my work, but it's the work of a group of great collaborators. And so I have here in the center Betsy Arnold, who is a professor at the University of Arizona in Tucson, Jim Dalling and Adam Davis, who are professors at the University of Illinois at Urbana-Champaign, and Carolina Sarmiento, who is associated at the University of South Florida. And so this has been a long-term collaboration with this great group of scientists. And one of the major questions that we have been asking ourselves in the last decade or so is to understand how seeds defend themselves in the soil when they land in the soil. But let me just back up a little bit and talk about tropical diversity. So if you travel to the tropics and you see a snapshot of the tropics, you will see a picture like this one, right? A lot of diversity, a lot of different crowns, different species. And so this tremendous diversity in terms of richness and composition has to be maintained. And that's a question that ecologists have been asking themselves for a long time. And there are different hypotheses and different theories to explain how we maintain, or tropical forests maintain, this tremendous diversity in terms of species composition, species diversity. One of those is density-dependent regulation by natural enemies. And so we know, at least, that at least in part of this tremendous diversity of tropical trees in tropical forests is the result of interactions between those tropical trees and their natural enemies. And so those natural enemies can be fungal pathogens. And today I will be talking about those fungal pathogens, but they can also be granivores. They can also be herbivores, right? And so at different plant stages, those natural enemies will be really important. And studying those interactions between the natural enemies and those trees can give us a lot of answers about understanding that high diversity in the tropics. And so in a cartoonish way, if we take this density-dependent regulation by natural enemies, what we are assuming is that if we have a mother plant, if we have a parental tree here in the middle of the tropical forest, the offspring of that parental tree will have lower survival under the crown of the parental tree than seedlings or saplings away from that parental tree. And so that result that has been tested empirically over and over again in different systems, different places in the tropics and also elsewhere, assumes some degree of host specificity. So those natural enemies killing those seedlings, seeds, or saplings below the crown of the parental tree need to be host specific, or at least have some sort of host specificity to have this pattern of lower survival under the crown and higher survival away from the crown of that parental tree. And so this concept of host specificity will be really relevant later on in this talk. But studying those processes by which microbial antagonists generate those empirical results that we have been documenting over and over again has been challenging, and has been challenging for tropical ecologists for several reasons. One of those reasons is that ecologists have been working mainly at this level of complexity. So from seedlings to saplings, then to understand the template for adult trees. But seedlings and saplings are encountering a very wide diversity of enemies: herbivores, granivores, pathogens, and all at once. And one of the things that I hope I will be able to convince you at the end of this talk is that seeds are a great model to study those density-dependent regulation by natural enemies in a way that is more manageable, and more manageable for empirical and experimental studies. But seeds are not only great models, they are also very important. So they are critical components of plant reproduction and plant fitness. And if we go to tropical forest, we have pioneers that can produce seeds that will, after they are ripe and they are dispersed and they land into the soil, they can integrate this thing that we call the soil seed bank, right? And so those are seeds that will lay and stay in the ground for different periods of time. And while there, they survive in these environments that are not only microbially rich, but also that has a lot of variation in terms of the physical environment. So, you know, you name it, in the tropics, wet season, dry season, so on and so forth. And what is remarkable is that some of those seeds can be persistent for a long, long time. So Dalling and Brown, over a decade ago, published this really great paper on American Naturalists, in which they found that at least for several species in BCI, in the middle of the Panama Canal, those seeds can be viable in the ground even for decades, even over 30 years. And so they used carbon dating to establish the age of those seeds. Then they germinated them and they carbon dated those seed coats, and they determined that they can persist viable there for even decades. And that's remarkable. If you think that some of those seeds are only a few millimeters long and they land in those places that are rich in terms of microbes and are very often wet, and so conditions don't seem to be ideal for bee there. And while there, those seeds interact with a wide variety of microorganisms. And so those microorganisms, we know that they could have positive or negative effects on our seeds, and so they can interact with them. And one of the things that we were really interested, or actually my postdoc advisor, Jim Dalling, was interested a few decades ago, is what are those seeds in the soil bank defended against? What is the major driver of seed mortality in the soil seed bank? And so let me get at that answer with these two charts. So what we have in these two charts is, on the y-axis, we have seed germination, and then we have two different species of trees. On the top, we have a species of Myconia, Melostomataceae, and on the bottom, we have a species of Cecropia and Urticaceae. And then we have seeds that were buried underneath the crown of the conspecific and 30 meters away from the crown of that conspecific. And then we have controls, so seeds that were not treated, plots that were not treated at all, and those are highlighted here in white. And we have plots that were treated with fungicide. So fungicide was applied. And let me just get to the first result, which is just looking at the controls. So what we can see for Myconia, but also for Cecropia, is that seeds buried only 30 meters away from the crown are surviving 2 to 3 times more than seeds underneath the crowns of conspecifics. So this is empirical data showing that density dependence is happening there for seeds. So they are dying more often if they land under the crown of a conspecific. Then the second result that is relevant here is the fungicide treatment. So seeds that were, or plots that were treated with fungicide germinated way better than controls, either beneath the crown of the conspecific or 30 meters away. So the take-home message of this result is that most probably those seeds must be defended against fungi, because fungi are the major factor killing at those seeds in the soil. And so this study was great, and it was really informative, and we can check the density dependence empirical results in the field. But this approach has some limitations. While in the ground, we didn't know much about how those seeds interact with the soil fungi, right? And so when I get to this project, one of the first questions that we ask is, how are those seed-associated fungal communities structured, right? So a purely descriptive way to understand those seed-associated fungal communities. And then a following up question is that if we can describe how those seed-associated fungal communities are structured, can we just name a little bit the function of those fungi? What are those fungi infecting seeds doing? Are they killing them? Are they just living there in a symbiosis? What are they actually doing? So to answer these two questions, we implemented this seed boreal experiment in BCI. And so we collected tons of seeds. And when I'm saying tons of seeds, it's literally tons of seeds. We buried over 150,000 seeds of 18 different species. And we buried them for different durations here in this little island called Barro Colorado Island in the middle of the Panama Canal. So this is Panama, we have South America here, and then Central America, okay? And so those seeds were buried in the ground for different durations, and they were buried in those little seed bags that really look like tiny party favors. And we extracted them little by little in time, okay? And so with those seeds that were extracted in the lab, we surface sterilized them, and then each particular seed was cut in half, and one half was using in a culture, and the other half was used to assess seed viability. Using this colorimetric test, the name of that test is tetrasolium, but names are not that important here. But the important thing is that for each particular seed, we knew if the seed was dead or alive at the time of retrieval. And then the other half went into this pipeline in which we culture that seed in media, and we had at the end fungal and bacteria growth. Today, I will only be talking about fungi, but bacteria, I believe, are also important in the system, but that's for another day. And so when we had pure cultures of those things that were able to colonize seeds, we Sanger sequenced them using ITS and LSU just to give them IDs. And so today I will be giving, for this first initial part of the talk, results out of 9 different species of tropical seeds. And here we have some names if you are interested on taxonomy. And seeds that were buried for up to 12 months in the ground in 5 different locations in BCI. And I will be presenting data from over 1,400 different fungal isolates that belong to over 200 different species of fungi. And so species of fungi were defined here as OTUs, defined at 99% similarity. So I will use species of fungi or OTUs along this talk. And so let me go to the first result. And so remember that the first thing that we wanted to characterize here was the seed-associated fungal communities. We use 4 different variables to associate those communities. And so we describe a seed-associated fungal community using plant species. So we used 9 different plant species in this experiment. Boreal duration, how much time those seeds were in the ground. And we have 4 different boreal durations. Seeds were buried for a month, 3 months, 6 months, and 12 months. Boreal location, we buried those seeds in 5 different common gardens in BCI. And seed viability status. So seeds can be dead or alive at the time of retrieval. And here we have the first set of results. So what we have in this chart here is an ordination. This is a non-metric multidimensional scaling analysis. And each point in this ordination is a seed-associated fungal community. So 2 points that are close to each other are two seed-associated fungal communities that are more similar to each other than 2 points that are farther apart from each other. And so the first thing that we can see here, and I decided to color those seed-associated fungal communities by plant species ID. So different plant species have different degrees of blue in this ordination. And so what we can see is that different species have different communities. You know, like this community is different to this one, and this one is very different to this one. But I know some of those results are still a little bit messy. So let me get at the same result using a different analysis. And so what we have here is a variance partitioning analysis using the same data, seed-associated fungal communities. And we have our 4 variables: plant species ID, boreal duration, remember 4 different boreal durations, garden location, 5 different garden locations in BCI, and seed viability status, dead or alive. And what is remarkable here is that over a quarter of the total variance is explained by plant species ID, only by plant species ID. But the other 3 variables together, that explain less than 3% of the variance. And so these 3 variables seem not that important to explain at how those seed-associated fungal communities are structured, and plant species ID, at least from the 4 different variables that we measure, seems the most important one. But let me just also highlight that there is a lot of variation that we are not able to explain in this model. But the first take-home message of this part of the talk is that plant species ID seems to be really important when we want to characterize those seed-associated fungal communities. Now that we know that, we can go to another question, and that is how seeds defend themselves in the soil. And so if we have different communities of fungi associated with different species of plants, we can assume that those seeds have different filters that will filter which fungi are able to enter and colonize those seeds. And so using this theory of plant defenses, we can also think that seeds must be defended using physical defenses, for example, very thick seed coats or impermeable seed coats, so physical defenses in general, or chemical defenses. And so investment will be here to synthesize or to allocate chemical compounds to deter granivores or pathogens, right? But we also know that, that defenses are costly. So we don't expect seeds to invest heavily in physical defenses and at the same time in chemical defenses. So we could have 2 different scenarios here that are not completely mutually exclusive. But one scenario is that as defenses are predicted to trade off against one another because resources are not infinite, and so we can expect this trade-off between physical defenses and chemical defenses. But we can also have this scenario in which we have defense syndromes, in which life history of those species will tend to cluster together. And so we have groups of species that tend to defend themselves in similar ways using similar strategies. And so we will have clusters of species defending themselves in similar ways, right? And then, of course, those syndromes can trade off against one another. So this is why I said at the beginning that these 2 are not completely mutually exclusive. But at least we have these 2 ways to understand how seeds allocate defenses, right? Or energy to defend themselves. And so we tested that framework. And what I have here is a linear discriminant analysis. So this is similar to an ordination. Each point in this analysis is one species. I included 16 different species of seeds in this analysis. And I included 4 different traits related to physical defenses and 2 chemical traits related to chemical defenses. And I will not enter into much detail about the data set, but you can ask me questions afterwards if you are interested. And what we found, the result that we found is that we have those groups of species that tend to group together, okay? And so I colored those groups here in red, green, and blue. And what is really interesting is that then when we look at the seed ecology of those species, those groups correlate perfectly well with seed dormancy. And so seeds can be dormant or non-dormant. And if seeds are dormant, they can be physically dormant. The two major types of dormancy in the tropics can be physically dormant. So those are impermeable seeds that need to break that impermeability to then germinate, or they can be physiologically dormant. And so they will need some cascade of chemical reactions to break dormancy to then germinate, or they can be quiescent or non-dormant at all. So ready to germinate as soon as they land into the ground and conditions are adequate for germination. And so this result was great because we were able to link these two processes, seed dormancy and seed defenses, that before our paper was published, no one was really putting these two things and these two concepts together. And we argue that seeds have these dormancy defense syndromes to defend themselves in the soil. And so right now that we know that plant species ID are really important at explaining seed-associated fungal communities, but also that we expect that species share traits to defend themselves, what happens with genera that are rich in diversity? And so we have that over and over again in the tropics. There are rich genera that, if what I just show is true, we can expect that those seeds will be defending themselves in similar ways. And so the question here was, if the pathogen-mediated coexistence hypothesis provides that general mechanism for the maintenance of diversity, we still expect some sort of differentiation in those soil-borne microorganisms that are able to colonize congeners, right? Otherwise, our findings will not be standing alone. And so we tested that hypothesis. And how we did that? Well, we used 4 different species of seeds. We used 3 congeners, so 3 species of Cecropia, and a species that has the same ecology of those 3, but is phylogenetically distant, Jacaranda copaya. And why those 4 different species? Well, because the seed ecology of those species is really interesting. So in this chart, those species will land into the non-dormant quiescent species. So those species are species that are not investing heavily in terms of physical defenses or chemical defenses, and are also permeable. And so we think that those were good examples of species in which microorganisms, and fungal in particular, will be important at understanding seed demography. And so we used the same setup of boreal experiments in BCI, but the addition here is that we buried those seeds for 30 months rather than a year. So 2.5 years. And we recovered them also little by little in time. And here the results. And so again, our seed-associated fungal communities were defined using those 4 different variables, the same as the first example that I showed. And the great result that we found is that even for congeners, so these 3 species of Cecropia and Jacaranda copaya here, we still find that plant species ID is the most important variable at explaining those seed-associated fungal communities. And if we go to this variance partitioning analysis, plant species ID explains over 30% of the variance. And again, the other 3 variables explain only about 6% of that variance. And so this is important because remember that we included 3 species that we assume that tend to be defended in similar ways. And even with those similar defense traits, they still have different susceptibilities to fungal infection. Just to highlight a little more this result, we did this pairwise comparison analysis, and again, this boreal duration, boreal location, and viability when we compare those communities coming from the same, let's say, boreal duration against different boreal durations. I guess, yeah, the battery of this is off, but that's okay. So these white bars and gray bars are very similar, but when we look at the genus comparison or the plant species comparison, which is same genus, so Cecropia versus Jacaranda, or same plant versus different plant, we have this huge difference between those communities. So again, showing that even if congeners tend to defend themselves in similar ways, there is a huge filter imposed by plant species ID. But remember that at the beginning of this talk, I started saying that density-dependent regulation by natural enemies is important, and we assume some sort of host specificity. And so our initial guess is, okay, if host specificity is important, if we look at how are those interactions playing out in our system, we expect some sort of host specificity. And the result is no, we don't find that. So what I have here in this network is on the bottom, we have a different species of plant. So our 3 species of Cecropia and Jacaranda. In green, we have seeds that were viable at the time of retrieval. In yellow, we have seeds that were dead at the time of retrieval. And on the top, we have the 8 fungal OTUs or fungal species that were more common in our data set. And what we see is that there are many lines crossing to each other. So there is no host specificity. So the next question was, how can we just reconcile that absence of pathogen host specificity with those results that empirically show over and over again that density dependence is happening in nature, right? And so in order to answer or to reconcile this idea, we implemented this inoculation experiment in which what we did was that we inoculated 7 different species of trees or seeds with 4 different fungal isolates. So those fungal isolates, remember that we have cultures. So those were fungi that were cultured from our seeds, okay? And we also used controls. So those were seeds that only seed on media, but they were not inoculated with any fungal strain. And then we measure germination using germination experiments to determine the outcome of those infections. And so let me walk you through the results. And so here what I have, and remember that we used 7 different species in this experiment, but for a time question, I will only presenting data for 2 of them. So we have here on the left Ficus insipida, and on the right Cecropia insignis. And on the y-axis, we have the proportion of viable seeds. That proportion was corrected by controls. So if this bar is equal to 1, that means that the seeds that were inoculated with a fungal strain germinated or survived equally well than controls. If that bar is lower than 1, that means that the inoculation killed somehow the seeds. And we used 4 different fungal isolates. And here we have names for the ones interested on fungal taxonomy. And so let me show the results for Ficus insipida. What we see is that different isolates, so all the isolates were able to infect those seeds in the inoculation experiment. That's the first experiment. But then the second result, the first result, sorry. But the second result is that different isolates do different things. In this particular case, isolate 2, Lacodiplodia, and isolate 4, Abionectria, killed over 80 to 90% of those seeds in the inoculation experiment. But isolate 1 and isolate 3 only killed about 20% of those seeds in the inoculation experiment. So different isolates do different things to those seed species. And then when we and that was true for Ficus insipida, it was also true for Cecropia insignis. But then when we compare those two different species of seeds, what we see is that, for example, isolate 2 that was killing over 80–85% of the seeds of Ficus insipida only kill about 25–30% of seeds of Cecropia insignis. And if we move to isolate 3, this was the most pathogenic isolate for Cecropia insignis and only killed about 20% of the seeds of Ficus insipida. So the take-home message here is that there is this very strong interaction between fungal isolate and plant species ID, right? And so let me just show that result with a particular example from our data set. So we have here this species of Lacodiplodia that was able to colonize 3 species. So remember, we have 9 species in the data set, and this species of Lacodiplodia was only able to colonize 3 of those 9 species, and that's why plant species ID is really important at explaining those seed-associated fungal communities. But what is remarkable is that this species of fungi was only recovered from dead seeds of Anonas spraguei. But we found that fungi also, or that fungus also on Ficus insipida and Cochlospermum vitifolium, 2 other species of seeds. And so the take-home message of this part of the talk and the inoculation experiment is that seed mortality is not only reflecting this ability of fungi to colonize the host, which is usually called in the literature as host affinity, but the outcome or the impact of that interaction can be host specific. And we call that in this paper functional specificity. And by putting this functional specificity in the literature, we think that at least we are able to reconcile this absence of host specificity in terms of interactions, but we have host specificity in terms of function. But one of the great things about working with nature and working with empirical data in nature is that nature is complex. And so it's difficult to really describe nature in good detail. And one of the things that we were also able to find is that so far I have been telling you this story of fungi colonizing seeds, and those fungi are coming from the soil, so soil-borne fungi. Justin Schaffer came into the group, and he actually found that in some of our fungi, there are bacteria living inside those hyphae. And so there is this endohyphal bacteria. And what is really cool about that is that you can treat those fungi with antibiotics. And so you can curate them and have the fungus with and without the bacteria. And we know how to inoculate those into the seeds. And so we can inoculate those seeds with the fungus, harboring the bacteria or not harboring the bacteria, and then see the outcome of those infections. And this is exactly what Justin did. And here is just one of the several results that he found. So for Cecropial Angipes, he inoculated those seeds with this particular fungus, this horrible name, with this particular fungus without the bacteria, and that particular fungus, including the endohyphal bacteria. And the results are the following. So here on the y-axis, we have the proportion of seeds that germinated. This line is just the controls. So numbers below this line will mean seed mortality, and numbers similar or close to this line will mean that seeds inoculated with those fungi are surviving as well as seeds not treated with any inoculation. And what we saw is that when the seeds were inoculated with the fungus without the bacteria, that fungus was really pathogenic. But when the seeds were inoculated without that bacteria, then the fungus is not pathogenic at all. And so that adds another layer of complexity. But what is cool is that we can say that context dependency also reflects that effect of endohyphal bacteria. So this tripartite interaction can be also important at understanding the outcome of those interactions or those fungal infections. And so to sum up, I hope that I was able to show you that plant host species is really important at explaining those seed-associated fungal communities in tropical forest, in those seeds that land in the soil seed bank. But also that not every fungus is able to colonize every species of seed. And this is why we have this plant species ID that is really important at explaining those seed-associated fungal communities. But a single fungus can colonize several hosts. And so in other words, a single fungus can be polyphagous in a way. That natural enemies such as seed pathogens can discriminate even at lower levels of phylogenetic relationships, so even at congeneric levels. And that is important at understanding seed susceptibility to fungal infection. That there is this very strong interaction between the plant species and the fungal species, and that those interactions can shape seed mortality and seed demography. And that pathogens and soil-borne pathogens can be really important at understanding this promotion of species coexistence in tropical rich forests in terms of species. And with that, I thank you all for your attention, and I will be willing to answer questions. And of course, this is not only my work, but the work of a great team of collaborators and a lot of people that help us in the lab, in the field, in Panama, in Illinois, in Arizona, and right now in Florida. So thank you again.
Thank you so much, Dr. Zalamea, for a really fascinating talk. We'll now take questions. Chris.
Such a cool topic, such really fascinating ecology and fungal experiments. Really neat. Thank you. The one question I wanted to ask first was right at the end of the talk, the result about the endohyphal bacteria. So why is that? Like, why is it that you take bacteria away and they become more, the fungi become more bad for the?
So that's
Can I repeat the question?
Sure.
All right. So Chris Solomon was asking in the experiment where you removed the endohyphal bacteria, why do you get a different effect on seed germination than when you have it within the hyphae?
Yeah, that's a great question for which I don't have a great answer. And so we don't know. And that's the real answer that I can give you today. We don't know, but we know that, you know, microbes in general, they are dependent on the physical environment, and they are dependent on the chemistry of the physical environment. And one of the things that we can probably imagine is that fungi, when they are colonized by those bacteria, maybe the bacteria or the fungus or the plant is producing some immune responses that can make the interaction less pathogenic, but we really don't know. But we think that at least in part, the answer is the language in which those interactions are happening, which I believe is just chemistry. But that's a really, really rudimental answer to a really great question that we don't know.
Peter.
Also enjoyed your talk tremendously. I had a similar question. So the microbial interactions with the seeds, there's probably interact with lots of other things like soil chemistry right now, soil chemistry. And here at the Carrot Institute, there was a lot of work on small animals eating seeds. They planted a bunch of seedlings, and the next day they were all gone because mice or voles had eaten them all. And seems that could keep you busy for a long time. So have you thought about like other things and how that interacts with the microbial, and how important are these microbial things versus other things that affect seeds? And I guess the other thing is, how does it also affect the plant to just make a lot of seeds? You know, they're over. So I guess the question of how does this microbial stuff experience, it has to fit in the bigger picture of the seeds.
So Peter Grafman's asking, how do other factors like seed predation and other things fit into the big picture of microbes affecting seeds? Yes.
Yeah, so that's a great question, and I have several things to say. One of the things is that, for example, in some of our, not some, in all of our experiments, we intentionally decided to eliminate one factor that is happening in nature, and is seed dispersal by animals, right? And so that is, for example, a frugivore, a bird or a primate eating this fruit, and then the seed passing through the gut of that animal, and then seeds landing in different places in the soil. We don't have that into account, but when seeds are passing through the gut of an animal eating those fruits, they are interacting already with this gut microbiome, right? So this is something that we intentionally decided to eliminate from our experiments, just to avoid another layer of complexity. And so, sure, there are many other layers of complexity, like, you know, the one that I just described, the one that you described, that is granivory. And I guess the thing is that, and I was talking with Sarah and others in the lab meeting early on, that we have different filters that happen at different scales in plant demography. And so, for example, seeds that will be persistent in the ground for a long time, I imagine they will be more susceptible to fungal pathogens than seeds that will germinate right away when they get into the soil, and then will create a seedling, for which maybe herbivores will be more important at that seedling stage, right? And so I guess all those factors are important, but the thing is that they just work as different funnels, different filters in this plant demography over time. And I don't know if that is a good answer to your question, but of course, our picture is just a snapshot of those seeds landing into the soil. And we try to avoid all the other complexities. So by putting those seeds in those party favors, we are avoiding granivores, or we are avoiding those other factors that can attack them, right? And so we only get a partial part of the whole picture. The whole picture, I'm afraid, is really complex. And so how to scale up from those individual seed fungal interactions to the big picture, we were talking about with Sarah early on, and I am convinced that we will need some modelers to be able to actually model those different funnels or those different filters that are actually happening at different stages in plant demography.
Yeah, and Charlene Tarsa raised that there's also soil invertebrates that could be one of these filters that might be important. Jane.
Super interesting work.
Thank you, Jane.
I have two questions. I'll focus this on one. Do you think that plants that are obligate mutualists within a microbial or microbial organism might not display that density dependent relationship because they are more likely to get that mutualist when they're near their
So
Can you repeat the question?
yeah, so Jane actually asked that there are some cases in which there are mutualisms that can be maintained through time. And I believe Jane is also talking about vertically transmitted microbes, right? In comparison to our system in which most of those microbes are horizontally transmitted, right? And so you are totally right. And we were discussing about priority effects early on. And what I think will happen is that on those vertically transmitted symbiosis or interactions in which mother plant will sort of like get birth with this good guy in, seed or whatever, priority effects will play a major role there. And I agree with you that my gut feeling and my expectation will be that those cases in which we know that there are vertically transmitted microorganisms, they will tend to have less density dependence than those systems in which we have a lot of horizontally transmitted microorganisms. So I agree with you. And that will be something cool to test. And so we have good examples, for example, in grasses, in which we have grasses that we know that they have these vertically transmitted microorganisms that they will keep for generations, or in orchids too. We also know cases that the seed will not germinate if there is no this particular mycorrhizae or things like that. And so that can be tested experimentally. Yeah, I'm not aware of any studies looking at that, but that will be cool to do.
We'll take a question online from Evan Gora. Evan says, very interesting talk. Do we know anything about the size of pathogenic fungi? In essence, are seeds typically colonized by hyphae from macrofungi that primarily exist outside the seed, or are they typically colonized by microfungi that primarily live inside the seed?
Yeah, so that's, by the way, hi, Evan. I'm really sad you're not here. But anyway, that's another story. So that's a great question. And so let me just start answering that question by saying that in our data set, we surface sterilize those seeds. And so the main rationale behind that surface sterilization is to get rid of microorganisms that are on the surface of the seed, but are not necessarily interacting with the embryo, right? And so all of our fungi are fungi that were able to colonize, not necessarily all the way to the embryo, but at least parts in the seed coat that the surface sterilization was not getting rid of those microorganisms. And so, yes, we have microfungi here. And I think, I don't remember the second part of the question, but I think that by doing that surface sterilization, we are just avoiding those microorganisms that can be in the surface, but not necessarily affecting, at least in a direct way, seed fate. But I feel that I'm missing part of the question.
Yeah, so he was just asking, so you're talking about microfungi that are inside of the seed?
Yeah, so definitely. So and the reason why is because I think that they are more important. They are not the only players, but they are more important at understanding seed fate.
Cool. Shannon?
Yep, that's an excellent question. And I was talking to Sarah and others early on that we are submitting this NSF proposal. And one of the things that we want to answer is that. So if you see all the results that I presented at a very small geographical scale, PCI, that is an island, that is small. Even if we buried seeds in different soils for different durations, it's still a very local scale. And so one of the things that we want to do, if this proposal is funded, and let's hope reviewers and NSF think that it's worth funding that proposal, is that we will set up boreal experiments along the Panama Canal. And so we will have a more large geographical scale, at least about 70, 80 kilometers distance between those boreal sites, in which we can at least get some sort of idea about how important geography is. And I believe geography will play a role there. And so another way to answer that question is we don't really know. We think those factors are important. We think geography, the physical environment. Another great thing about the Panama Canal watershed is that there is this very steep rainfall gradient in which it rains almost twice in the Caribbean side when compared to the Pacific side. And so we also have this tremendous variation in terms of rainfall. And we think that microbial communities in the soil will be affected by that. And of course, then those interactions between those soil-borne microorganisms and the seeds will be affected by those spatial scales that we are not taking into account so far in our experiments. But yeah, great question. Again, another great question with not a great answer.
One more question. Michelle?
Awesome talk. Yeah.
Thank you, Michelle.
A little bit at like not being lethal for fungi is maybe a good thing. Like, and so to what extent is it good for like the plants to tolerate like a diversity of fungi? Do they play a role in defensible or I'm just curious about the evolutionary like conservation.
Yeah.
So I'll just repeat. So Michelle Wong is asking, is there a benefit to the plant of having multiple fungi? And is there an evolutionary arms race?
So So that's a great question, Michelle. And one of the really nice things of being here is that I will be able to talk to Shannon, and unfortunately Barbara is not here, but I guess disease ecology people, they know a lot about that, and we don't know much about it. But my idea in the system is that it cannot be a good strategy just to infect your host and wipe it out. You need to have some sort of like reservoir in the system. And so by being polyphagous, those fungi are most probably just being able to play with those in sort of like a long-term scale, playing with those abundances of trees at the landscape level by being more or less lethal. And so being less lethal in a particular one-to-one interaction can just function as a reservoir for that particular interaction. But that's something that I'm just speculating about. And my feeling is that disease people are ages more, that they know more about those disease dynamics, that we are just grasping the tip of the iceberg with those studies as the ones that I presented today. And so that's why I'm super looking forward for the meeting with Shannon later on today to talk a little bit about that.
Wonderful. Well, thank you so much, Dr. Zalamea.
Thank you,
And
Sarah.
let's thank him once again for a really wonderful talk.